Tumor-reactive monoclonal antibodies have been studied for decades, and they're really making a difference clinically. There are a number of ways that they can have effects on the way tumor cells grow or don't grow. Some of these pathways can involve specific causation of death through death receptors on the tumor. Some of them can be blocking growth factor signals on tumors. The two pathways that have been of most interest to immunologists are the effector functions of antibodies themselves. One of these includes the activation of complement, which can cause a protein-induced osmotic lysis of cells. But this doesn't seem to be very important in the way most monoclonals might be binding to and killing human tumor cells. More important than this is the involvement of cells that have Fc receptors, receptors that allow them to see tumor cells when they're coated with an antibody. These cells then will bind to the antibody that's stuck to the tumor and either cross-link it, causing separate biological reactions in the cancer cell, or more often, activating immune effector mechanisms of those cells, like natural killer cells or neutrophils or macrophages. Particularly the natural killer cells are the ones that we and many other immunologists are interested in, because they have the capability of using multiple pathways to then destroy those tumor cells that have been coated with antibodies. This works in vitro, it works in mice, and if you do your clinical trials in a way that simulates in patients the conditions that worked in animals, one can see really dramatic anti-tumor effects in patients. So if the pathway that we're interested in for monoclonal antibodies that bind to tumors involves these different effector cells of the immune system, one could hypothesize that anything you could do to augment the function of those effector cells would help in your monoclonal antibody-mediated anti-tumor effects. So if one is interested in the use of macrophages that are using their Fc receptors to bind to antibody-coated tumor cells, one might imagine that those macrophages or their similarly derived dendritic cells might function better at processing antigen and presenting it if those cells were stimulated with something that might help them do a better job of that. One such approach is the use of agonist anti-CD40 antibody, which is being used really as a vaccine adjuvant in many settings, preclinically, with soon-to-be tested clinical applications. The approach we're more interested in is looking at the effector functions of cells like natural killer cells that mediate this antibody-dependent cell-mediated cytotoxicity. When they do so, their Fc receptors bind to the antibody stuck to the tumor cells, and then those natural killer cells form an activated immune synapse that binds to the surface of the tumor cell, and they then use granzyme and Fas in order to destroy the tumor cell. If those natural killer cells are activated either before they encounter the monoclonal antibody or at the same time, one can get those natural killer cells to function far more potently in destroying tumor cells. This can happen in vitro, and it's happening in mouse models, and now there's growing clinical data that this can happen there as well. The approaches that are being used include molecules like IL-2 that we and others have studied for a long time. More recently, molecules like interleukin-15 both can activate natural killer cells to be far more potent at mediating ADCC. A more novel approach that's being pursued particularly by the team at Stanford, including Hobart Court and Ron Levy, is that of anti-CD137 antibody. This is a molecule that's expressed on activated natural killer cells when their Fc receptors are being activated. And by tickling that CD137, these natural killer cells mediate far better ADCC. So all these approaches are working in vitro and are now moving into clinical testing. So the session at this year's conference that we're participating in this afternoon is focusing on the concept of immunocytokines. This is really a category of bifunctional antibodies where one of the end of the antibody recognizes something that's specific on the tumor cell, and the other end of this molecule recognizes something on the effector cells. There's a few ways these have been formulated. One involves an antibody that recognizes the tumor, linked either genetically or chemically to an antibody that recognizes a triggering structure on your effector cell, like the CD3 structure on T cells, or the Fc receptor on natural killer cells. Such molecules as blinatumomab, recently approved by the FDA, has used this process to engage T cells to bind to the tumor cell, trigger them, and get them to be able to kill. A number of structures are now being pursued, some in the clinical setting, some have many years to go before they get there. The immunocytokine concept, really conceived by Stephen Gillies, who we've been collaborating with since 1992, took the initial tumor-reactive antibodies he was studying and had been using and linked them genetically to cytokines, such as interleukin-2. The idea here is that the anti-tumor antibody will bind to the surface of the tumor cell it's specific for and coat that tumor cell now with IL-2, because the IL-2 is on the end of that antibody. So IL-2 is normally thought of as a soluble growth factor that's present in soluble plasma or serum. But now you've got cells that are coated with it on the surface, and it allows effector cells like T cells or natural killer cells that have IL-2 receptors to use their IL-2 receptors to actually recognize the tumor directly, to bind to the tumor cells, to polarize those IL-2 receptors to the tumor cell and create an active immune synapse that turns on the effector cell, causes a resultant killing reaction, and recruits other cells into the area. And at least in vivo, this is a potent way of generating long-lasting adaptive immunity. While preclinical work, either in vitro or in animal models, can set up the experimental conditions to take best advantage of the experimental immunotherapeutic you're looking at, these experiments really need to simulate the clinical conditions that would be faced by the clinical oncologist. For that reason, it's important to look at where in the course of a patient's care one might use these agents. So putting a tumor into an animal and studying it only a day or two later is a very different in vivo setting than taking a patient who has had a cancer developing for many months and might have a kilogram of cancer in them at the time that they're diagnosed, and when in that patient's clinical course of care is the right time to use these immunotherapeutics. For many of these antibody-mediated therapeutics, including bifunctional antibodies or immunocytokines, particularly when dealing with solid tumors, there are a number of factors that allow these agents to work best when you're dealing with smaller amounts of cancer. Larger cancers make it hard for these large molecules to penetrate into the interstitial spaces between the tumor cells, so they don't penetrate well. In addition, the larger the tumor is, the more immunosuppressive pathways are turned on, not only T regulatory cells, but myeloid-derived suppressor cells. So for that reason, both in our animal models and in our clinical testing, we're focusing on trying to use these approaches for patients that would have smaller amounts of cancer. That translates to patients who were initially diagnosed with cancer and then given their initial upfront treatment, be it surgery or radiation therapy or chemotherapy, or in the case of neuroblastoma, the combination of all three of those, to put those patients into either remission or a near remission state. And in that setting, have smaller amounts of cancer that are resistant to those standard treatments, like chemotherapy. And that's when implementing something like immunotherapy seems to make the most difference. That's where we're focusing and where we think molecules like immunocytokines or bifunctional antibodies might have the greatest possible influence. So we're seeing pretty striking clinical activity using first-generation monoclonal antibodies or some genetically engineered variants of these, such as the humanized or human antibodies. In the first-generation immunocytokines or bifunctional antibodies, we're seeing activity as well. And the clinical trials are really based on mouse or other animal models where efficacy is seen to try and translate what works in a mouse into the clinical setting. Now that we've got clinical data, we're seeing what unique hurdles are present in the clinical setting. We're learning about issues that pertain to pharmacokinetics of these molecules and how that needs to be considered as the next generation molecules are created. We're seeing toxicities that are identified in patients that were hard to imagine or to predict from the animal work. And so it's possible to generate the next generation immunocytokines or bifunctional antibodies to try and do that. So for example, our colleague Steve Gillies is now working with next generation immunocytokines that are putting the IL-2 into a different place on the monoclonal antibody in order to facilitate the interaction of IL-2 with those receptors that we think are most important in the anti-tumor effects, namely the alpha-beta gamma receptors, while trying to interfere partially in the interactions with those receptors that might be responsible for the IL-2 related toxicity, namely the beta gamma receptors. So by modifying the position of the IL-2, by modifying how it's attached to the antibody through genetic linkage, and by modifying other parts of the antibody molecule, the hope is to get better pharmacokinetics, to get a larger area under the curve, to get less toxicity from these molecules so one can use higher doses and take advantage of the dose-dependent anti-tumor effects, and to be able to give these molecules in a more clinically expedient way, such as subcutaneous dosing on an every few day basis, as opposed to needing to use constant infusion delivery, as is currently the case with some of these smaller molecules. So it's really a combination of lab-based immune design linked to preclinical testing in animals to design the initial clinical trials, taking the next step and moving it into clinical testing and seeing both activity and hopefully efficacy, but then going back to the laboratory and saying, now what can we do to improve these molecules to make them even work better so that we can have broad application? Our goal in all of this is to generate molecules that are potent in destroying tumor, that can be stored on the shelf of any hospital pharmacy around the world, so that this kind of high-tech technology could really be available to cancer patients no matter where they're getting treated anywhere in the world. I really think it's not a dream. I think we're really in the process of getting there.